So we have two photos in total. Two are from the kind of graphical modeling approaches. The first one is by Alberto Roverato, giving us an alternative to partial correlation measures.
Thank you very much. Okay. Well, the title of the talk is the networked partial correlation and its application to the analysis of genetic interactions. So I would like to start just explaining what the genetic interaction is. So when biologists want to understand the function of a gene, what we typically do, they knock out the gene, so they silence it, and in this way they produce the so-called single gene mutant. And since this gene cannot express itself, then this way we can understand the function of the gene somehow. Now, what happens if you consider an organism like a yeast, the yeast Saccharomyces cerevisiae, for instance? It has about 6,000 genes. Now, you can silence this gene, one gene, and then look at the overall thickness of the yeast of the organism. And it is very common that you silence a gene and nothing happens. So the cell is as healthy as before, essentially. And what is somehow strange is that this happens for most of the genes. So apparently no gene is really necessary. A possible explanation for that is that there exist the so-called buffering relationships between genes. So there are pair of genes, and when one gene is not working anymore, the other counterbalance the expression of a silenced gene. And so for this reason, what now biologists do, they produce the so-called double mutant organisms. So they silence a pair of genes and to see what happens, essentially. And what they do in the lab, essentially, is this. They silence a gene and check the fitness of the organism. They silence another gene independently and check the fitness. And then they are able to predict the fitness of a double mutant in the case there is no buffering relationship. Then they actually silence both genes, check the fitness, and if the observed fitness is different from the predicted fitness, then they decide that there is a genetic interaction. Of course, this kind of experiment requires the silencing of all possible pairs of genes, and this is not feasible in practice. They will show you that they did it for the yeast, for the 6,000 genes of a yeast, but if you consider the human genome, it is really impossible to consider every pair of genes. So what is necessary is to try to identify candidate genetic interactions by using computational tools, essentially. And there are a number of possible covariates that can be used in this case, and biologists use protein function and localization, homology relationships, and protein-protein interactions, and other things. But a role here is also played by the expression level of genes, because it is known, the biologists know, that when there is a buffering relationship, it is also always true that the two genes co-express, and essentially there is a positive association between the two genes. And well, these computational models the biologists use, essentially they consider very, very simple quantities such Pearson correlation, Spearman correlation, partial correlation. I mean, there is a lot of room for improvement here, definitely. Now, from my point of view, if I want to look at the co-expression of genes, I always think of a co-expression network and based on partial correlations, essentially. So for me, I consider P genes, one from 1 to P, and XV is the random vector of the expression of the genes. So sigma is the variance and covariance matrix. Its inverse is the concentration or precision matrix, denoted by K. And the partial correlation between two genes, the expression of two genes, U and V, given overmanning genes, is obtained by normalizing the partial covariance, or it can be obtained by scaling the off-diagonal element of a concentration matrix in this way, minus K UV over the square root of a product of the diagonal entries of a matrix. Now, it is obvious that if I want a zero partial correlation, I can check for a zero partial covariance, and it is equivalent to a zero of diagonal entry of a concentration matrix. And when the vector is normally distributed, then a zero partial correlation is equivalent to a conditional independence of two variables given all the remaining variables. But here I only look at linear relationships. I don't need the normal distribution. And then we already seen in this workshop how to associate a graph to a concentration matrix. So there is one vertex for every variable, for every gene, and an edge is missing when there is a corresponding zero in the concentration matrix. And when, I mean, in the Gaussian case, these are the covariance selection models or Gaussian graphical models. Now, in a co-expression network like this, one edge denotes the presence of an association between the two genes. And of course, this is a candidate genetic interaction. But we also need at this point to decide somehow the strength of the edge. And so we need to put a weight on the edge. And what is typically done, the most straightforward way to proceed, is to associate a partial correlation, because we see the partial correlation is zero when the edge is missing, and so it is a nice quantity to associate. It's very common to see a matrix with points, and darker points means a stronger partial correlation, and maybe a white point is a zero partial correlation. But we didn't want to stop here, and we checked for other possible ways to be associated to the edges of a graph. And we looked at, I mean, there is a theory I'm going to show you in a moment that associates a weight to every path of an undirected graph, and we decided to look at this kind of thing. So if we have a graph like this, we have one gen X and another gene Y, and for instance, a path is a sequence of vertices that are joined by an edge from X to Y. So for instance, this one is a path X1 to Y, and this one is another path X1, 2, 4, 5, Y, and we can go on this way to find the third path, a fourth path, a fifth path, and a sixth path. So there are 6 possible paths between X and Y in these graphs. And Jones and West, in a paper published in Biometrica 2005, they gave this nice way to decompose the covariance between the two genes. So sigma xj is the covariance between the expression levels of X and Y, and this is computed at the sum. The sum is for all the paths. This capital pi is the collection of all paths between X and Y. So there are 6 paths, for example, in the previous example. And, every path is associated with a weight that is a function of a path and of a various covariance matrix. And the expression for the weight is this one. So it's a function of the edges. These are the edges of a path, and this is the function. So when associating weights to our graph, we decided to look at this kind of decomposition. And in the paper of John and West, they wrote this sentence, and essentially there is a way to decompose the covariance between two vertices of a graph in directed graph, And this lead to path analysis, to the theory of path analysis that developed by Wright in 1921. And they they thought that this decomposition they provided for undirected graph could be an equivalent of path analysis in the undirected case. And in fact, when I read this paper a few years ago, I thought we were right. I mean, that this was very nice decomposition of a covariance of a path, of a graph. But then, after more than 10 years, when I look again at this thing, I was not able to find any single application of this theory. So there is really the idea is that nobody used it or has used it so far. And mainly, I was really surprised. And I think that the reason why this theory was not applied afterwards is because the interpretation of the weights associated to the path is very unclear. And for instance, when we tried to use them, we had the problem to normalize path, to be able to compare path and maybe to be able to compare path with different endpoints. So we started looking at the possible interpretation, and well, of course, we started from the very simplest case. The very simplest case is a single edge of a graph, because an edge is a path. So we looked at an edge, the weight of the edge, and just a matter of writing it in a different way. And if you write it in a different way, you will see that the edge, the weight of the edge xy of sigma is this ratio. The numerator is a partial covariance. It's the covariance of x, y, given the remaining variables. And down here, there is a function of this rho, and this rho is a vector correlation coefficient. It's a vector correlation coefficient between the two variables, the endpoint of the edge, and all the remaining variables. So the vector correlation coefficient is a classical measure of association between vectors. It was introduced in 1965 by Roseboom, but in fact it is based on a vector alienation coefficient that was introduced by Hotelling in 8 years ago. This is the way you can compute the vector correlation coefficient between XA and XB, but in fact it is equivalent to Wilks lambda for testing the dependence of normal distribution. I mean, the sampling version of this is a Wilks lambda, and it can be computed by using canonical correlations. I mean, it's a very classical measure of association. There's nothing new here. But this way of writing the weight of an edge is very useful for interpretation of the meaning of this weight. And it is clear that we have the numerator is the partial covariance. And the partial covariance is a measure of the strength of the association, but it is computed adjusting for all the remaining variables in the network. So essentially, it is a strength of the edge, but does not keep into account the possible connections of this edge with other vertices in the graph. So it's sort of outer measure of association. Clearly, the weight and the partial covariance have the same sign, and then, of course, when one is 0, the other is 0. But more generally, the weight is always larger. The weight is larger than the partial covariance in absolute value apart from the sign. In this sense, the weight is an inflated partial covariance, and the inflating factor is the denominator. The inflating factor is based on this vector correlation. So the stronger is the linear association between the two vertices and all the remaining variables in the graph, the more the partial covariance is inflated. And for this reason, we decided to call this quantity the networked partial covariance, because it is a partial covariance sort of embedded in the graph, right? And well, okay, when the two vertices are not connected, then the weight and are not connected with the rest of the network, then the weight and the partial covariance have the same value. So this also provides a very natural way to normalize the weight of an edge, because we can normalize the partial covariance, and in this way we obtain a network partial correlation. So just to give a very toy example of what's going on here, we can, for instance, consider this graph here, and a various covariance matrix such that all the partial correlations, pairwise partial correlations, are equal to 0.4. This gives a valid variance covariance matrix, positively defined, and these are the weights associated to the edges. And of course, all the edges are on an equal footing, they have same value. If we now compute the network partial correlation, since we start from identical partial correlations, where we see that, for instance, this edge and this edge have the same value because this is disconnected from the graph, but the other are not, they are larger and they are inflated, and the value of the network partial correlation here depends on the number of connections of the edge with the other. So, for instance, here there is a 1.7, and it's the largest value because it is the most connected edge. And so we thought that this quantity might be more suitable in the case, for instance, of genetic interactions, because in a genetic interaction, it is necessary that the two genes have a positive association, in the sense that we want positive partial correlation. So we want positive network partial correlation, essentially. But then our intuition suggested also that one pair of genes have a strong effect on the fitness of the organism if these two genes are strongly connected with all the other or other important genes in the network. So we thought that this quantity might be more appropriate, or we could work better in the prediction of genetic interactions. I don't think I have much time to get into this, but these networked partial covariances seem to be very natural quantities to use in this kind of graphs. Sometimes are even more natural than partial covariances or partial correlations, and this is the reason why. So typically, what we do, when we compute partial covariance, a partial correlation, or network partial covariance, we are not observing all the possible variables. There are always some unobserved variables, and so essentially what we do, we compute these quantities on a marginal distribution. So we have a quantity computed in the marginal distribution, and it is always useful to try to understand what happens, how this quantity can be interpreted with respect to the variables that we have marginalized over. And the network partial covariance has a very nice rule to update its value, and its value can be updated by looking at the path that disappear as a consequence of marginalization, and so it is very clear how a path can affect the network partial covariance under marginalization. And with such a nice connection with, for instance, partial covariance, I don't think that is there. So, they seem to be very natural quantities to be considered in this case. Anyway, let's take a look at the problem of genetic interactions. So we consider this data, and these are data for the GIST Saccharomyces cerevisiae. It's a sample of 112 observations, and there are about 6,000 genes. And we use this data set because the way the experiment was conducted allows to investigate closely this pathway. This is a well-known pathway. It is the leucine pathway. It's a pathway that produces leucine in the GIST. And in this pathway, there is a metabolic intermediate called alpha IPM. It's very important intermediate in the process, and there are two genes. They are denoted by LEU4 and LEU9, and this is a known genetic interaction. As far as we know, it is a unique genetic interaction known in this pathway. And what happens essentially if you can silence LEU4 and nothing happens, silence LEU9 and nothing happens, and if you silence both genes, then there is a reduction in this metabolic intermediate, and the production of leucine decreases drastically, and so there is a problem with the fitness of an organism. Now, well, this is a representation of this pathway, a possible graph associated with this pathway. But here, what we have, we simply computed the Pearson correlation, the network partial correlation, the partial correlation, and here again the network partial correlation. And what was turned out very, and we liked it a lot, is that the Pearson correlation was not able to highlight the role of these two pair of variables, and the partial correlation didn't do it either. But the network partial correlation gave the highest value to the unique genetic interaction engine. So, it was nice. Other values, other pairs of genes with high value of network partial correlations are all upstream with respect to the alpha IPM. So, it is possible that there are pairs of genes with a possible strong impact of the fitness of a gene because they are upstream of this alpha IPM. Second, we carried out a genome-wide analysis, so considering all the genes of this data set. And in order to have a benchmark, to have a gold standard to compare our values of a network partial correlation, we consider this paper, and in this paper published in Nature Methods, what they do, they in the lab check the genetic interaction of almost every pair of genes in the gist, and they compute a quantity that is called SGA score, and when this SGA score is negative and large, then there is an evidence of a genetic interaction. So we downloaded these SGA values and just checked whether there was a possible correlation between our network partial correlations and these SGA values. And so just looking down here to these two panels. So for instance, we consider the partial correlation here and this SGA score, we see that there is a positive association and the R square is 0.07. When we change the, and we go to the network partial correlations, we have a stronger association. I have to say that the improvement is not huge. We go from 0.07 to 0.16. It's not huge, but it is significant. And you have to think that we are using gene expression data to predict genetic interactions. And genetic interactions are only one of many features that affect the expression of a gene. So it is unrealistic to expect something better from this kind of analysis. But it is encouraging, at least it's an improvement with respect to the use of partial correlations. The next step, we try to understand whether this kind of interpretation is possible also for an arbitrary path, for a general path, not for a single edge of a graph. And this is the approach we followed. So if we consider a subset of V, or subset of the genes, and then so we split the vector XA in XA and X into XA and XA bar, we can compute the linear regression of XA on XA bar, consider the residual, and this is the variance covariance of the residual, and its inverse is the submatrix of K indexed by A. Now what happens is that the graph associated with this matrix is just the subgraph induced by A. So for instance, if for me A bar is the vertex phi, the subgraph induced is the same subgraph where I have to remove the vertex 5 and its edges. So at this point, if we look at one edge, one path in this graph like this, then I can compute two weights for this path. I can compute the full path weight using sigma and the partial path weight using the partial covariance matrix. And these partial path weights can be used to decompose the partial covariance over the path in these subgraphs, but it's not so important. What is more useful is to write the path weights in this way. I can write the full path weight as a ratio. Here, the numerator is the partial path weight, and the denominator is the function, is this usual function of the vector correlation coefficient between xa and xa bar. And clearly, only the numerator depends on the path. The denominator depends on a, and that's it. As before, the two weights have the same sign, and again, the full weight is larger than the partial weight. And this is, these are all linear relationships. What happens here is the same as linear regression. When you increase the number of covariates, the variance of the residuals decreases, right? So this is exactly the same. So when we increase the number of the covariates, so of these A bar, we have a smaller weight. So if by increasing the number of covariates we can reduce the value of weight, it makes sense at this point to say, well, if I'm looking at this path here, well, I can simply consider the vertices of a path, and everything else is a covariate, and so write something like this. So P are the vertices of a graph, and I can compute this ratio where I have here the weight, the partial weight for where P are the vertices of a graph. So this is a way of decomposing the full weight in such a way that the numerator is as small as possible. Then, forgetting for a moment this kind of representation, second problem is that of a normalization of a path weight. Well, again, playing a little bit with these quantities, it's possible to show that one can write the weight in this way. The weight is the covariance multiplied by this phi pi r. R is the scale concentration matrix, so essentially it is a matrix of partial correlations, apart from the sign that is negative. And so this is the way one can write it, this phi thing here. Now, what happens? This phi is a normalized quantity, because we sum it over the path between x and y, it is equal to 1. And then it is scale invariant. It is scale invariant in the sense that we can replace R by delta, where delta is the diagonal matrix for rank, and we obtain the same quantity. So in fact, we can, instead of R, we could put K, and it would be the same. So this is a normalized quantity, and for instance, it also shows that if I compute this weight, and instead of sigma, I put C, that is the correlation matrix, what I obtain the correlation by this phi. This phi is always there because it is normalized, doesn't change. So we can normalize the pathway, computed it as a function of the correlation of a scale covariance matrix, or we can just normalize the covariance here, and we obtain the same quantity. But what is more useful for me is to take this kind of factorization and then this ratio here and put them together. And we obtain a form like this. We can write the weight of a path, and this is a network partial covariance, and this is this phi computed on the set matrix RPP. And the network partial covariance here only depends on the vertices of a path, not on the edges of a path. And this quantity depends on the path, and as before, it is a normalized quantity, and its sum is equal to 1, but I have to consider the sum over the path that involve only vertices in P. So it's a subset of a path between x and y, and this gives value 1. Now, if I look at a path like this, I can, for instance, remove the vertex 1 and still have a path, because x2, y is a path, and this is a path involving a subset of the vertices of a path. But for instance, this path here cannot be shortened. It's not possible to remove edges without destroying. I mean, there are no subpaths of this one. And the same is, for instance, down here. Again, this is a path that cannot be shortened. When a path cannot be shortened in this way, well, this phi quantity is equal to 1. So clearly, an edge of a graph is a path that cannot be shortened. But this is more general. When we have a path with this property, then its weight is a networked partial covariance. Things become more complex when, in this case, for instance, so when I have, when I look at this graph, in fact, I should look together to this path and to this path, because this is subpath. And so in this case, I have two phi values, one for the first path, one for the other. The sum of these two phi values is equal to 1. And so interpretation is, I mean, it is easy to say that you can do many things, but I'm not so convinced. So I have not still completely understood the behavior of this thing now. When the sign is positive for the two phi here, then of course we are giving the part of a partial toward partial covariance to one path, and the proportion to one path, and the rest of partial covariance to the other path. When we don't have the same sign, some phi are positive, some phi are negative, so it's less clear how these pathways can be interpreted, and this I'm still working on that, I have to say. So concluding remarks. Well, what we have done in this work, we have hopefully given some useful insight in the interpretation and properties of these pathways, and hopefully we will open the way to possible applications of these quantities that I think may be very useful in practice. We have already shown an example where these network partial correlation can be useful. For instance, this problem of genetic interaction, they explain more variability than other quantities used so far. Completely open problem, the inference for these quantities, and of course, in this application to genetic interaction, it's a very early stage, and we have to consider more sophisticated model, including other quantities to predict the genetic interactions and see how network partial correlation actually work in these full models. That's all. Thank you for your attention.
I think that if all the partial correlations are non-negative, if they're positive, which is the same as to say that the multivariate distribution is totally positive of order 2, then I believe these pathways, or the right version of the pathways, can be interpreted as probabilities of a random walk that go from x to y, and every pathway is really the probability that a random walk will do exactly that way. And I think that might be an interesting way of trying to understand what goes on with the various paths.
Yes, of course. One thing that I notice when I work on this stuff, it is always tempting to think to give a direction to a path. So to think that you can, I mean, the real problem in the interpretation here is that you have always to think that it's a symmetric relationship.
Yes, but if you have a random walk, which this of course is time reversible, so this is so you have a random walk. You start it in x, and then you kill it the first time it hits y.
Right.
Okay, that is a time reversible process. Does not look different from the one that starts at y and is killed the first time it hits x.
Okay.
So X and Y are absorbing states for that random molecule.
Thank you.
And then I believe that if the partial correlations are non-negative, which means that of course they would be strictly positive, otherwise you would have zeros. But if they're non-negative, strictly positive, then they can be interpreted as probabilities that this is exactly the way you would want to go.
Thank you.
And I think that might be an interesting way of understanding these ones. If they have different signs, some correlations are negative or something, then I think it gets quite difficult. And that's also when you get all the paradoxes.
These kind of network partial correlations, they kind of get the interpretation of some kind of centrality measure of the edge. Have you looked at kind of...
Yeah, we
So rather than a vertex, but you actually just the edge.
Yes, because when we give this example where all partial correlations are equal to 0.4, I mean they have this kind of interpretation, but they well, first of all, we can compute it without a graph. So, and second, there is these partial covariance in the numerator that is important as well. So. We call them network partial correlation, but in fact, when we looked at the association between, for instance, x, y, and all the remaining variables. So we might have a very strong association with just one edge, or maybe the same association with 10 edges. We cannot distinguish between this kind of thing. You see, we cannot see whether the association is with just one other gene or with many other genes. So in this sense, we cannot associate it to the edges really of a graph, to a centrality measure computed in terms of paths or this kind of thing.
But the network partial correlations are associated to edges, right? Existing edges, or to any pair of
to the strength, to the global strength of the edges. So you don't know, when.
Yes, but it is always an edge. It's not like two pairs of vertices that are, because they have partial correlation 0. So if they're not in the partial correlation graph, so the edge is not there, then the partial correlation is 0 and the network partial correlation is 0.
Yeah. Okay. Yeah.
I need to understand how you compute the Pearson correlations. If you knock two genes and you see that they work together, but how do you measure? What is the measure?
No, but the spatial data we used, there's no knockout. I mean, this is the point. If in the lab you can carry out this experiment, but you knock out and check for genetic interaction. But the point is that we want to try to predict genetic interaction from this state.
How do you measure? What is this Pearson partial correlation? How is it measured? What data you have?
We have a matrix of expression data. We have a matrix of expression data. Okay, we have a matrix like this. We have about 6,000 genes, 112 samples, and of course, we can compute a variance covariance matrix. Of course, there is a problem that this is not positive definite, so there is some problems with the estimation of partial correlation. Imagine that this matrix is positive definite, but it is not. You can just take the inverse and scale it, and you obtain the partial correlations. I mean, the sample version of the partial correlation. In this case, it's a little bit more complex. We have to use some method for the case where the number of observation is smaller than and the number of observation is smaller than the number of genes. But I mean, the way we
correlation in gene expression between the different genes. Is that right?
So it is, yes, it is the partial correlation. I mean, these are expression levels of the genes, and so you can. I mean,
it's not clear how it's correlated to knocking out two of them.
No, this is the point. We want to see. I mean, when you have a genetic interaction, it is known that these genes have a positive association, right? So we are trying to use this to identify genes with a positive association to see whether they can predict.
What is the biological meaning of a path?
What is the biological meaning of a graph?
No,
It's more
of a graph.
yeah, no, I know, but Ernst, in his talk a couple of days ago, explained it very well, that it's an ideal thing, right? Because here we consider two genes and how essentially we have two genes, but two genes don't interact directly. So one gene produces RNA and then proteins, and then they only interact indirectly. This is a representation of the association structure, essentially. There is no, we represented some graphs, for example, where you have a transcriptional association. One gene produces a transcription factor that activates another gene, so there are this kind of mechanisms. Here we only look at associations.